Welcome to our cellular energetics debate. Tonight we have candidates Donald Triphosphate and Hillary Catabolism. They will be discussing the issue of metabolism.
We have known that photosynthesis and cellular respiration have very similar processes, and
Mr. Triphosphate, you may begin the discussion with explaining why your process, photosynthesis, is better than Mrs. Catabolism's.
I'll start with carbon dioxide, water, and simple sunlight. And by putting the cell to work, I will store the energy necessary to make our cell great again.
thank you, Mr. Triphosphate. Ms. Catabolism, it is now your turn to explain your argument as to why process cellular respiration is better than Mr. Triphosphate.
Alright, thank you. Well, although our processes are very similar, I would say that let's keep in mind that they are opposite reactions. I'll start with glucose and oxygen, and I will be able to yield carbon dioxide and water to generate enough ATP, not just for the cell, but for the entire body. My catabolic breakdown pathways and anabolic buildup pathways are always ready for work.
Thank you, Mrs. Catabolism.
Mr. Triphosphate, how do you plan to aid the plant cells in dark times?
You know the nice thing about me? I'm rich. I mean, really rich. And with how much glucose I have, if the cell needs 600 ATP, I can make it for him. I say the cell doesn't have access to light. Listen, I've been in plenty of tough decisions, and I can use my experience with the carbon cycle to make sure the cell survives. I'm a photoautotrope, and I'm not dependent on taking glucose from other organisms. I can give stability the cell needs.
Thank you, Mr. Triphosphate. Mrs. Catabolism, would you care to briefly explain what you have already done for the cell in your last few years' work?
Well, gladly, my friend. You see, the systematic process known as cell respiration, it starts in the mitochondria. As good old heterotrophs we are, we must get the energy received from ingesting other organisms. Now keep that in mind. As part of my job in the last few years, I worked very hard to ensure that energy coupling occurred, and each outward reaction, exergonic, and each inward reaction, endergonic, is present. I also worked very hard to make sure to send my very best oxidizing and reducing agents out to aid catabolic pathways in the production of ATP. These agents do just as they're told. They either oxidize, lose electrons from a substance, or reduce, add electrons to another. Interestingly, these atoms are not directly transferred to oxygen. They're passed through an electron carrier, a coenzyme called NAD+, that when it is oxidized, and when it is not oxidized or reduced, it is NADH. Well, I would say that is the most important thing that I've been working on these last few years.
Wonderful. How do you feel about the biodiversity within your cell, Mr. Triphosphate?
Let's be frank here. I love the biodiversity of photoautotrophs, but C4 is obviously the most useful. Just look at what they do. Corn and sugarcane both come from C4 plants' ability to produce sugar in low CO2 levels with their mesophyll and bundle sheet cells. You want to get your morning sugar fix? There's your answer. Anyway, I'm not saying the other cells are completely useless. I think they're special for their own unique abilities. Qualities. C3 plants only use Ubisco. For those of you who don't know, RUBP, which is a 5-carbon sugar that gets attached to CO2 molecule to incorporate it into the carbon cycle, carboxylase, which catalyzes the said bounding of RUBP to CO2 to fix CO2. So you know, not really the best since Ubisco also fixes O2 in this way, but if they have the right boost of high CO2 levels, they do a great job at carbon fixation. And then there's my 50 little cams. As a side note, I normally don't like political correctness, but they prefer to be called cressulation acid metabolism plants after the process they use to store CO2. So while they may be slackers in plant cells, I value them despite their inferiority.
Mrs. Catabolism, I heard a rumor that there are various stages of cellular respiration that is a process. Can you confirm or deny these rumors?
Well, I'll not just confirm them, but I will also do you one better, and I will explain them. Glycolysis begins to degrade by breaking down glucose into 2 molecules of a compound called pyruvate. These pyruvates enter and are oxidized into a compound called acetyl-CoA, which enters a cycle called the Krebs cycle, which forms a small amount of ATP directly from phosphate level phosphorylation. And then finally, the compound will go through oxidative phosphorylation, making ATP from ADP, empowering the electron transport chain to start it all over again. Most of the remaining electrons carriers between the oxygen and compound called ubiquity are proteins called cytochromes.
Interesting. Miscatabolism. Now, Mr. Triphosphate, there's a misconception regarding photosynthesis that states that plants make oxygen for humans and humans give plants back carbon dioxide. Is this true or not?
Of course it's true, but it's also slightly wrong. You see, both cell types undergo cellular respiration, so plants don't really depend on humans to have CO2. However, they do have a net generation of O2 to make excess that humans can use.
This is to both candidates. Can you explain your implementation of ATP synthase and how it will help our cells?
Gladly. First, I'll explain a big picture idea. A proton motive force, that is a force that generates potential energy by going against its hydrogen ion concentration gradient. This movement is very important because our ATP synthase enzyme will need it to make ATP from ADP. It will pull electrons outside of the cell, then, by chemiosmosis, pull them back in from their reserve to make ATP for the cell. You know, it's really quite interesting.
Mr. Triphosphate?
Yes, it does sound very interesting. But I think you fail to see the weaknesses in your system. You're always so greedy, and this is no exception. You want to consume so much energy, but with my system, I have the sun do the work for me. Let me break it down for you so you can understand. Plant cells all have chloroplasts. Those chloroplasts consist of two other outer membranes, which are filled with a fluid called the stoma. In this fluid, the thylakoids are stacked into grana. The thylakoids do a wonderful thing. They are what make the sun do work for them. Most importantly, they have built a very special wall, like walls, to keep hydrogen ions in the thylakoid space, which is the fluid inside the thylakoid, and out of the stroma. The thylakoid membrane, I like to call it a wall too, has two photosystems in it, which are complex molecules. The first one is in the process in photosystem 2, which uses solar energy to strip a water molecule of its hydrogen atoms, which would then become ions, which is called photolysis. The material that gathers the sun energy is what is called chlorophyll. High energy electrons are also stripped from the water and gathered into the primary acceptor, which uses the cytochrome complex, which is also called the electron transport chain, to take electrons to photosystem 1, which produces ATP at the same time. It's very efficient. The photosystem 1 uses light energy, which, by the way, you can't get any greener, folks, to reduce NADP plus to NADPH in a process called photophosphorylation. Chemiosmosis is simultaneously, it's very advanced, synchronized technology, very new and very exciting, used with a membrane or wall molecule named ATP synthase, which uses the diffusion of hydrogen ions across the thylakoid membrane to power the phosphorylation of ADP to ATP. These molecules are used to power the Calvin cycle, which produces 3-glycideraldehyde 3-phosphate molecules, also known as G3P. That combines into a glucose, all perfectly green energy.
One last question. What are your plans for aerobic reactions, processes needing air, and anaerobic reactions, processes not needing air, Mrs. Catabolism?
Well, this one a little shorter. Regime is for alcoholic fermentation to be strictly anaerobic process, stand on its own from cell respiration. For lactic acid fermentation to be able to freely be made into cheese and yogurt. My dream is for yeast and bacteria to make enough ATP that they can call themselves true facultative anaerobes. Keep in mind our processes are exact opposites, but I will assure you, mine is much more efficient. Vote for me, and all this may come true. Hillary Catabolism for America's cellular responses.
That's all for today, folks. We have heard both arguments from Mr. Triphosphate and Ms. Catabolism. Make sure to vote for the best process, and remember that the choice that you make can either catabolize or anabolize our cells.